To start, I hope you all slept well. Today is an exciting day. We get to hear what's inside and how it's connected to the outside. The playlist that you've been hearing is something I hope you'll add to. There are two public playlists on Spotify. One is called Energetic Mind and one is called Mellow Mind. And so if you have Spotify, you can add your favorite song. Just no hard rock, but then we get to hear it. So this morning we're going to have Elisa Hamlet introduce Steve. It's going to be brief because we're already running late. And Steve Horvath flew up from LA just for us, and he has to fly back today. So if you want to say hi to him, this is your chance. So we're going to turn the Zoom on once we, where did Steve go? Good morning, everyone with us watching virtually. We're so happy you joined us. We had slight technical issues yesterday. I hope you weren't too frustrated. And so we're just finished editing the amazing Terry Moffat talk. And so that is, you will get recordings. We have gotten many, many emails saying, will we get recordings? You will get all the recordings. Eke Dubois has made an amazing, beautiful landing page. So it's one-stop shopping. You're going to get the link later today, and you will have access to the videos as they pop up within an hour or so after the talk. Thank you for joining us.
Thanks, Alyssa. Good morning, everybody. I'm Elisa Hamlet. I'm a postdoc at UCSF, lucky to be working with Alyssa and some other folks here. And I'm very pleased to introduce Steve Horvath, who's a professor of genetics and biostatistics at UCLA Public Health. When you think of epigenetic clock and biological aging, you should think of Steve, who is a pioneer and leader in this area. We are very lucky to have him with us today. Thank you very much. Please welcome Steve Horvath.
Thank you. It's a real pleasure to be here. I will talk today about causes and consequences of epigenetic clocks. This is really a magical place. I had the best breakfast ever and beautiful redwoods, but one thing was missing, and this is we needed a statistical formula, and here it is. This is the statistical definition of an epigenetic clock. An epigenetic clock is fundamentally a multivariate regression model. There's a dependent variable, which could be chronological age, it could be mortality risk, it could be lifespan. But then there are these covariates. It could be millions of cytosines. And when we build epigenetic clocks, we use a mathematical optimization method to develop the most accurate clocks. So the algorithm selects cytosines and forms a linear combination. But whenever you have a multivariate regression model, you then face the challenge of interpreting it. And this has been a challenge to the entire field. What is the biological interpretation of these epigenetic clocks? And many people have worked on it, and we all use a variety of tools, genetic studies, various interventional studies, for example, in vitro studies where we expose cells to certain reagents. We, of course, use epidemiological studies, longitudinal studies, and more recently, even comparative biological studies. And I'll talk about all of these studies today to shed light on the meaning of epigenetic clocks. I want to introduce different generations of epigenetic clocks, which are really defined by the outcome, meaning the dependent variable in the regression model. The first generation epigenetic clocks estimated chronological age very accurately. The second generation of epigenetic clocks aim to go beyond it and estimate mortality risk or morbidity risk, the elusive biological age. And today I will also present the third generation of epigenetic clocks, which estimate not age, not mortality risk, but rather what I call relative age. And relative to what? Relative to different species. So these are now clocks that apply to mice and rats and also humans. And they do that by adjusting for life history traits. So here, for example, I show you a human rat clock. And just to be clear, this is again a single regression model. And the dependent variable is this relative age, which is defined as the ratio of chronologic age divided by the maximum lifespan of the species. For example, the maximum lifespan of rats is 3.8 years, maximum lifespan of humans, 122 years. And so if you form this ratio, you end up with a number between 0 and 1. And this ratio, it's rather unsophisticated. It's in certain ways the simplest thing you can think of to put different species at the same level, right? But anyways, this very simple operation works. And today I will show you plenty of these kinds of figures where in the left panel, you see the rat samples in orange and the green samples are human samples. And the x-axis shows you the relative age. So this is a number between 0 and 1. And the y-axis is a cross-validation estimate. So clearly, this clock aligns these different species. The right panel that you will see today is always the respective species. So these are now literally different rat tissue samples. And you see a very high correlation, well above 0.93. So remarkably, there is a single formula that allows us to really estimate age in these very different species, human and rats. And you should ask yourself, what is it good for? Well, of course, translational studies. You want to translate your studies in human epidemiology to a rat model or vice versa. And so this is now feasible with these clocks. We also built a similar human sheep clock. So this now uses ear samples or blood samples from sheep. Left panel is again relative age, two species combined. Right panel shows you that this clock has a remarkably high accuracy in sheep samples, correlation 0.98. And so why did I study sheep? Well, I was interested in castration, the effect of castration in rams. And we did find that castrated rams have younger ear samples. And I know all the men in the audience right now think, I want to have younger ears too. So on Monday I'm in the clinic. But before you do that, I want to caution you that we studied the effect of castration on blood samples and there was no effect. And I looked at castration in cats and dogs and horses. So blood will not be rejuvenated by that. And that brings me to a point. A lot of interventions really have a cell type specific effect. For example, later I will talk about rapamycin has an effect on keratinocytes, but not on blood. Similarly, anything related to hormones typically affects keratinocytes. And that may be relevant for some of you who collect samples because buccal swabs, saliva, that samples that capture keratinocytes may be more informative when you're interested in topics surrounding hormones. We have a human dog clock that was trained on 93 different dog breeds. So we can evaluate stress in your dog and in the owner of the dog. If you are more of a cat person, we have a dual species clock for humans and cats, blood samples from cats, very accurate. We have a human pig clock. Why? Many people study mini pigs as a model. So we can now do exciting animal studies in a mini pig model, and they probably will translate to humans. Why? Because it looks at conserved cytosines, identical cytosines in both species. We analyzed 33 different primate species, and we developed a primate clock. It's remarkably accurate. For any blood sample, or often we have multiple tissues, liver, kidney, brain samples from these different primates. So we have these primate clocks that apply to all primates, apparently. But this now presents here the most ambitious part of this project is to build pan-mammalian clock. So these clocks now apply to all mammalian species, including marsupials. We have samples from kangaroos or possum. So a distant species from us, bats and so on. And the lower panel shows you again this relative age estimate, the ratio of age divided by maximum lifespan, and we can clearly, accurately predict relative age in all of these species. Here I show you how the pan-mammalian clock performs in humans. The upper left panel shows you that this pan-mammalian clock has an accuracy of a correlation 0.95. In other words, it's as accurate as the first generation epigenetic clocks, the pan-tissue clocks we had. But now this clock is one regression model, and it applies to all species. And the other panels show you results in mice, in dolphins, in bats, in elephants, in dogs. So remarkably accurate measurements. Again, why do we do it? Because there was always this problem of translating findings from one model organism to another. And the hope is that these third generation clocks will increase the probability that, that results will translate from one species to another. And to provide an illustration of that, I want to describe a study of Down syndrome. So back in 2015, we already knew that a human Down syndrome was associated with greatly increased epigenetic age in both blood, but also in brain samples. That was 2015. And it took 6 years. And now we know that this finding translates to a mouse model. There's this TS65 Down syndrome model, which is which contains 2/3 of the genes that are orthologous to the human chromosome 21. And we looked at different tissues, liver, muscle, blood, and sure enough, we again see epigenetic age acceleration in that model. So I think this is kind of a prototype study of what could happen in the future. We have one finding in one species, and then we validate it in another species. But let's now ask this important question. Why can one use cytosine methylation to build, on the one hand, pan-tissue clocks, but on the other hand, pan-mammalian clocks? Stepping back, it's a miracle that one can even do it. Because if I asked you to build a dual species clock based on transcriptomic data, you would probably fail. I certainly failed at it. But similarly, even if I ask you to build a pan-tissue clock in mice, it's based on transcriptomic data or other omics data, I think people would have a very, very hard time. But with cytosine methylation, as I showed you, all of this is relatively straightforward. And why is that? to get at that, I show you here the results of an EBOS of age. So this is an epigenome-wide association study. We correlate cytosines with chronologic age in over 100 species. And we can see, well, which cytosines get hypermethylated with age in many, many species, which lose methylation with age. And to be precise, you could ask that question for each tissue type. You can say in brain tissue, which cytosines gain methylation, in liver, in muscle, in skin, in blood. And you form a Venn diagram and you say, all right, these cytosines that gain methylation in all these tissues, in all these species, what is known about it? And if you do a bioinformatics analysis, you will find that these cytosines tend to be located in bivalent chromatin, bivalent promoters. But the real story is target sites of so-called polycomb repressive complex 2. So the upper line on the right shows you p-values. These are p-values 10 to the minus 500 when it comes to this polycomb repressive complex 2 binding sites. So PRC2 is evolutionarily conserved. You can find polycomb proteins in mammals, in insects, and even fungi. PRC2 plays a role in maintenance of stem cell fates, X-chromosomal inactivation, imprinting. But I'm telling you, this is the smoking gun of pan-tissue and pan-mammalian clocks. That's why it's possible to build these. I want to now switch gears and talk about these famous hallmarks of aging that have been identified. And you can ask the question, well, which hallmarks of aging are related to epigenetic clocks or epigenetic aging effects? And here I show you a list, and that was really assembled by Ken Raj. And so cellular senescence, interestingly, is not really related. Same with telomere attrition, same with genomic instability. However, deregulated nutrient sensing, mitochondrial dysfunction, stem cell exhaustion, and altered cell-cell communication appears to be related to epigenetic aging. And biology is always complicated, but I will now show you some experiments that demonstrate these claims. And I start with altered cell-cell communication. Here I show you a rat study by Harold Katcher and Akshay Sanghavi, who developed some sort of a young plasma treatment. And so they administered that young plasma to rats. And we looked at various organs in these old rats. And what we found is that this young blood, in other words, these young communication molecules, they reversed epigenetic age by roughly a half in blood, in liver, in heart, not in the hypothalamus, but in all these other organs. So stunning age reversal effect, one of the strongest effects. Now I want to come to another hallmark of aging, nutrient sensing. And here the panel shows you studies in keratinocytes where the red dots show how methylation age increases as you passage the keratinocytes. Cumulative population doubling levels increase. The orange dots show you rapamycin-treated keratinocytes, and you see suddenly the aging stops, you know. But also, we had done many studies along those lines, you know. So rapamycin seems to affect keratinocytes. Another line of evidence about nutrient sensing comes from caloric restriction experiments in mice. There are over half a dozen papers that all demonstrate without a doubt, if you put a mouse on caloric restriction, you will slow the epigenetic age in liver samples. And then there are, of course, human sample studies, human epidemiological studies, and they have shown that, yes, BMI is correlated with epigenetic age acceleration in blood. The correlation is relatively weak, maybe 0.15. But in liver, the correlation is huge, maybe 0.4 or 0.5. So yes, all of that points to nutrient sensing. Another hallmark of aging, stem cell exhaustion. So here, the plots show you different cells collected from 4 different donors. And these are now keratinocytes, and the orange bars correspond to stem cells, keratinocyte stem cells. And the blue bars correspond to stem cell depleted keratinocytes. And what do you see? Well, stem cells exhibit substantially younger epigenetic age. So there is some sort of a connection to stem cells. It's a bit complicated because not all adult stem cells appear to have this kind of property. For example, hematopoietic stem cells don't seem to be much younger. However, there is a connection to stem cell biology, there's no doubt. Another hallmark of aging that's quite famous, mitochondrial function. And here Ken did an elegant experiment where he exposed neonatal fibroblasts to 2 types of agent. CCCP dissipates mitochondrial membrane potential. In certain ways, CCCP, which corresponds to the black dots, destroys the functioning of the mitochondria, and you see a great increase in epigenetic age. Conversely, if you administer bezafibrate, the orange dots, you see a decrease of epigenetic age, and this why bezafibrate increases mitochondrial biogenesis. So these experiments strongly suggest that mitochondrial function relates to epigenetic age. I'm hedging my language a little bit because we also had various mouse models where we knocked out critical genes for mitochondrial function, and we applied mouse clocks to these animal models. And then we often did not see epigenetic age acceleration, so it's complicated. Some experiments show an effect, others don't. Another hallmark of aging, genomic instability. You can induce that by X irradiation. And the long story short, X irradiation has no effect on epigenetic age, which suggests that genomic instability does not have an effect or is not related to epigenetic clocks. And Ken carried out very comprehensive studies. First of all, he administered acute irradiation, 20 gray, high dosage, but then no effect. Then he administered various regimens of chronic low-dose gamma irradiation, 1 milligray, 20 milligray, 50 milligray, different time points, pulsing, not just human cells, also mouse embryonic fibroblasts. So I would say Ken and actually several other labs have done these irradiation studies. There was never a connection with epigenetic aging, which is a bit frustrating, but it teaches us something about the biology. Now to the complicated topic of senescence. Here, the plot shows you different donors, 15 different donors, and these cells were now exposed to different ways of inducing cellular senescence. First of all, irradiation, then RAS oncogene expression, and replicative senescence. So the red dots are replicative senescent cells. And what do we see? The only way of inducing senescence that is associated with increased methylation age is replicative senescence. But why? Well, in order to achieve replicative senescence, you actually need to wait a while, right? So in this case, the cells were cultured for 6 months. And 6 months is enough time for methylation age to increase. By contrast, these fast ways of inducing senescence. The RAS oncogene irradiation did not lead to an increase of methylation age. And this brings me, when we talk about replicative senescence, this of course immediately brings us to another experiment. Use TET or telomerase immortalized cells. And here, these are now human endothelial cells, and the red dots show you these immortalized, age-TET immortalized cells. The x-axis is cumulative population doubling levels. And what do you see? Although these cells are immortalized, they will never become reach replicative senescence, but their methylation age increases, And this then explains the previous experiment, right? Why the replicative senescence was associated also with increased epigenetic age, because the time that passed led to an increase. And this brings me to another old finding. So this idea that TET immortalization does not stop the epigenetic clock. It brings me to another old and curious finding. Arke had carried out a genome-wide association studies of epigenetic aging rates in blood. Large study, GWAS study. We had methylation arrays, we had SNP samples. And we were asking, well, which SNPs are associated with what we call intrinsic epigenetic age? And we found the TET locus. And the right panel shows you the p-values, the Manhattan plot for the TET locus. The red dots are the p-values for intrinsic epigenetic age acceleration. And the blue dots are the p-values for SNPs that are associated with leukocyte telomere length. And what you see is perfect consistency. I mean, this is striking. The same TET region, the same everything is associated in GWAS of both biological clocks, telomere and epigenetic age. However, there was a very paradoxical finding. The genetic variants in the TET locus that were associated with longer telomeres, they were actually associated with this is a typo, accelerated epigenetic age acceleration, right? So if you won the genetic lottery and you inherited longer telomeres, then actually your epigenetic clock was accelerated, And but now we understand this biologic finding. Well, if your hematopoietic stem cells have longer telomeres, they in certain ways live longer, they can, time passes longer, therefore epigenetic age can increase in blood. I want to now come to a very important topic, which is the question, well, is cytosine methylation causal? Is cytosine methylation part of the machinery of aging, or is it a marker of aging similar to gray hair? And you can really answer that question in two different ways. There's strong arguments to say, no, cytosine methylation is not causal. And I would point to the following. Many invertebrates do not have cytosine methylation. C. elegans, Drosophila, these are very important model systems in aging research, but they simply don't have cytosine methylation. Conversely, though, they age, right? And also, a detailed bioinformatics analysis always show that many of the age-related cytosines do not seem to be associated with gene expression changes. However, on the plus side, there have been several exciting case studies where the authors showed that cytosine methylation does play a causal role. So David Sinclair recently showed that TET enzymes are needed for regeneration of cells. So if you use these OSK, the Yamanaka factors to reprogram axon regeneration, you do need these TET enzymes. It's kind of a smoking gun that TET enzymes play a role in regeneration. And there was another very nice paper by an independent group that looked at the ELOF-L2 gene. And if you are a methylation aficionado, you will know this ELOF-L2 gene. It's one of the top cytosines in human epidemiologic studies that is associated with increased age, whose cytosine methylation increases with age. And so this group showed that if you reverse ELOF-L2 promoter hypermethylation in vivo, in a mouse, the ELOF-L2 transcript was increased, and more importantly, you rescued age-related decline in visual function. So to me, this was a very nice study, arguing that yes, maybe cytosine methylation does play a causal role. I want to briefly switch gears. I want to talk about findings from the mammalian methylation consortium. So we have accumulated data from over 200 mammalian species. I think by now it's over 15,000 arrays, very large effort. And what I can tell you is you can use cytosine methylation to develop very accurate predictors of maximum lifespan in species. So again, maximum lifespan in humans is 122, in a mouse it's 4.8 years. For many species, the maximum lifespan is only inaccurately known, right? But you still take a rough estimate of the maximum lifespan. And despite all of that, you get correlations of 0.9. So it's, to me, remarkable that cytosine methylation captures that. I want to come to a challenge of the field of epigenetic clocks. And this is, we need to find clinical applications of epigenetic clocks. That's, I hope one of you will develop that. We have, of course, a biomarker that many of you know, Grim age. It's our best mortality risk estimator. It has been validated in many cross-sectional studies. And recently we developed a polygenic risk score. We have SNP markers that predict in certain ways Grim age. And this polygenic risk score for Grim age was significantly associated with parental longevity, in other words, longevity, and adiposity, even educational training, and so on. So you can use your SNP markers to evaluate epigenetic clocks. I wanted to give you one vignette about the potential clinical application of epigenetic clocks. And this brings me to what is known as clonal hematopoiesis of indeterminate potential. Another name is age-related clonal hematopoiesis. I will now use the name CHIP for this acronym. Now, CHIP is strongly associated with chronologic age. In other words, you gain mutations in cells that greatly increase your risk of leukemia. And so this CHIP is increased with an age-related increase of this risk factor is associated with various blood cancers, but also coronary heart disease, and of course, all-cause mortality. And there have been 2 studies that independently showed that yes, CHIP is actually increased with epigenetic age acceleration. And that's a very strong effect. I'll show you the results from one study that looked at methylation data and SNP marker data from 5,500 people. And we looked at 7 different epigenetic clocks. And really all epigenetic clocks showed epigenetic age acceleration in people who had mutations in these DNMT3A or TET2 genes. So these 2 genes are, mutations in these genes are associated with this CHIP and also increased risk of leukemia and also epigenetic age acceleration. I briefly mentioned CHIP can also be caused by mutations in DNA damage response genes. And interestingly, mutations in those genes, such as P53, were not associated with epigenetic age acceleration, only DNMT and TET2. And so why do I mention it? Because we have some evidence that if you link these two biomarkers, meaning epigenetic age acceleration, and combine it with the CHIP mutation phenotype, you can identify a population of patients at a particular high risk of all-cause mortality or heart disease. And you see in the lower panels, the purple curve, the y-axis shows you the risk of death. The purple curve are CHIP mutation carriers that also have positive epigenetic age acceleration, greatly increased risk, whereas the CHIP mutation carriers that did not have epigenetic age acceleration, they were in essence fine when it comes to mortality and heart disease. So I'll stop this presentation by reminding you of the main messages. We have found that cytosine methylation allows you to build pan-mammalian, pan-tissue clocks. Cytosine methylation allows you to estimate the maximum lifespan of species. The biologic story points to this polycomb repressive complex 2 associated gain of methylation with age in most tissues and species. I showed you compelling data that epigenetic clocks are linked to 4 hallmarks of aging: stem cell exhaustion, nutrient sensing, mitochondrial function, intercellular communication. Epigenetic age is distinct from telomere length and senescence. And epigenetic clocks are already being used in clinical and preclinical studies of anti-aging interventions. Here's my acknowledgement slide. I have to thank many people, and I'll stop with that. Thank you. Other questions?
We'll take questions at the mic,
Yeah.
and Elisa will bring us questions from our virtual audience.
Very interesting. Rob Lustig, UCSF. 2 questions, actually. First, you listed 7 different phenomena associated with cellular aging, but you left out autophagy, and I wondered if there was a reason why you did. It seems to be extraordinarily
just
important.
negligence.
Sorry?
Negligence.
Negligence.
No, I could have added it though. We had one mouse model of autophagy where one form of autophagy was knocked out, and I didn't really see an effect for that. However, I wasn't quite happy with the experiment. I was concerned. I didn't quite trust the data, you know. So I would say the jury is still out on autophagy. It really needs to be investigated, you know. And if you have an exciting experiment for autophagy, I would love to work with you, yeah, or anybody else. Yeah.
And the second question is semi-related. You mentioned mitochondrial dysfunction and suggesting mitochondrial biogenesis can reduce the rate of the epigenetic clock. So is it because of reactive oxygen species that have something to do with cytosine methylation? And do other things that increase mitochondrial biogenesis, for instance, exercise, for instance, ketogenic diet by increasing sirtuin 3, do those have the same effects on epigenetic clock advancement?
Yeah, we looked at SIRT3 knockout, you know, in hematopoietic stem cell. This was a collaboration with Danica Chen in a mouse model, and SIRT3 knockout did not seem to be associated with epigenetic age, you know. When it comes to exercise, there's a weak effect, you know. So if you study blood samples from thousands of people, you will see that there's a beneficial effect, you know. But the effect in blood, at least, is only moderate. But now we are doing some studies in rats, actually, where we exercise the rats and want to look at various organs, you know, and but these experiments are not yet finished. I also want to caution all of you about this claim about the connection to mitochondrial function, because some of you, Alyssa certainly has measures of mitochondrial function in from epidemiological studies. And if you correlate those measures with epigenetic age, the correlation was very weak, you know. What I showed here were really in vitro studies. That's where we saw striking results. Unfortunately, biology is just so complicated, you know. It always depends what tissue do I look at. Is it blood? Is it keratinocytes, you know? And so it's just complicated, yeah. The one thing I really trust is Down syndrome, you know. That's where I see it in all species, in all tissues, you know. Yeah, please.
Thank you, Steve. This question is from Elizabeth Blackburn, who's joining us virtually. And apologies if I get this wrong, but did mutations in uracil DNA glycosylase, UDG, superfamily in mammals, TDG, and MBD4 genes affect DNA methylation aging?
You know, I don't think I've looked at it, but it would be wonderful if Dr. Blackburn could email me and I'll put it on the list of mouse models. You know, I'm currently looking really at many mouse knockout studies. I have a list that I'm going through. You know, we've already looked at maybe 30 knockout mice or various transgenic mice, but we'll put it on the list. Yeah.
Thank you.
So you mentioned castration as one approach, but I guess I'm curious if you could talk a little bit about lifestyle factors or behavioral interventions. You know, a lot of us here, you know, study health behaviors in various forms, and whether that would hold promise, or which areas would hold promise for modulating epigenetic clocks.
Yeah, I'll start by saying by now I've turned into a total health nut, you know, so there were these happy days where I would eat salami 3 times a day, but those days are over. But so yeah, lifestyle is hugely important. But when it comes to these epigenetic clocks, it's actually a difficult question, because it comes to the question of what tissue should you look at, you know. What I can tell you is healthy diet, and now I'm talking about a measure of healthy diet, carotenoid levels, right? If you measure carotenoid levels in blood, then that measure is very much correlated with, for example, grim age in the expected way. If you eat vegetables, your grim age is reduced, and the correlation is quite strong. I want to say -0.4. But not for my original pan-tissue clock. It's really the second generation clocks, the mortality risk predictors that show it. The other thing I can tell you is smoking, huge effect in the wrong direction with grim age by definition. Now, when it comes to exercise, I feel we need better clocks, you know. So in my lab, we currently work on this, build a clock that responds to an exercise intervention. I have a doctoral student working on it, you know. I can't, if I trusted our clock, I would have shown it today, you know, I would call it work in progress. But then to me, it could be that exercise maybe has only a weak effect on blood. Maybe we need another readout. I think the new frontier is buccal swab, just other sources of DNA, you know. And all of us got tested with a nasal swab, you know. Why can't we collect nasal epithelium? And maybe it could be that this is suddenly far more responsive to various lifestyle factors. There have been very nice studies of post-traumatic stress disorders, and that showed that really long-term stress seems to be associated with a grim age acceleration, you know. Apparently short acute stress less so, you know. But This is not quite my field. Jude Carroll has shown a very nice study where she demonstrated that sleep disorders are associated with increased epigenetic age according to second-generation clocks. Okay, great. Well, I will be around.
Yes.
Bye-bye.
And
Thank you.
thank you, Steve. Steve, you are not only, brilliant and have changed the field of aging, bringing us these measures from, your background of deep mathematical modeling and engineering and your interest in aging. So it's just, feels like a real gift to the field and a new field that's emerging. And we're just so grateful. And Steve is also a phenomenally generous collaborator. So many of us here are collaborating with him. And I don't know how you have the bandwidth, but somehow there's a lot going on in this field, and you've been shepherding and curating it. So we actually have a 5-minute break before I'll introduce Dan Belsky. Thank you.